So we are very pleased to host today in our Bar-Ilan University Vision Science Seminar Professor Caroline Robertson from the Department of Psychological and Brain Sciences at Dartmouth. Professor Robertson did a BA in Neurobiology and Behavior at Columbia and then a PhD in Psychiatry at Cambridge University, where she was a Gates Cambridge Scholar working with Simon Baron-Cohen and an NIH Cambridge Fellow working with Chris Baker. She then continued to do a postdoc in neuroscience at Harvard and at the McGovern Institute for Brain Research at MIT, where she worked with Nancy Canwisher. After her postdoc, she joined the McGovern Institute for Brain Science at MIT as a research affiliate and also joined the Dartmouth College, where she's now a professor at the Department of Psychological and Brain Science. She received multiple awards and honors, including the Society for Neuroscience Janet Rosenberg Trubach Young Investigator Award, the NSF Career Award, the National Academy of Science Kavli Frontiers of Science Fellowship, the Brain and Behavior Research Foundation NARSA Young Investigator Award, and more. She publishes in top leading journals as Nature Reviews Neuroscience, Nature Neuroscience, Nature Communications, Current Biology, PNAS, Autism Research, and more. She's the head of the Robertson Lab at Dartmouth College, where she investigates perception, memory, and neurodiversity. Her lab studies the neuroscience of how people see, remember, and neurodiverge by embracing a wide range of experimental and computational techniques such as VR with eye tracking to study complex human behaviors in naturalistic settings, and neuroimaging as fMRI, MRS, EEG, and pharmacology and computational approaches to study the neurobiological mechanisms underlying these behaviors. Dr. Robertson is also the director of the Dartmouth Autism Research Initiative, which is the DARI, and her team seeks to better understand how people with autism see the world and to increase our scientific understanding of the neural basis of that experience. So without further ado, I'd like to welcome you, Caroline, to our seminar to hear about the exciting research that you are doing, and thanks so much for joining us.
Thank you so much. And yes, it's really an honor to be here. I was saying before we got started that I've watched many of the great talks in this talk series over the years, and mostly on YouTube because I tend not to be ready to go right at 8 a.m. East Coast time. But it's such an excellent series, and I'm glad to get to participate today. So my plan today is to share with you some work from my lab that primarily comes from the fMRI side of our work. And in particular, the last third of the talk is unpublished and ready for feedback. Everything is ready for feedback, but I'd really love to get questions along the way, and I'll stop a number of times along the way as well to ask if people have questions. So I'm planning to share with you some work today from our lab in which we use fMRI to understand how memory informs perception. To start, we're going to focus on the test bed of naturalistic scene understanding. When you look at this visual scene, you can immediately recognize it as an outdoor scene, a building, probably on a college campus. And of course, over the past few decades, we've developed a pretty good idea of how the visual system accomplishes visual scene recognition like this. Neurally, we've identified specialized areas in the high-level visual cortex that are causally linked to scene recognition. And behaviorally, we know that humans can perform many scene recognition tasks in the briefest of a glance, as little as 30 milliseconds. And computationally, we now have models of scene recognition that rival human levels of performance. But all that I've said so far leaves out an important part of my experience of this scene. Because for me, from the moment that scene appeared on the screen, it immediately situated me in the center of the Dartmouth campus, feeling the looming presence of our clock tower just over my right shoulder. This penumbra of memory is, of course, critical to our normal, everyday, natural experience of visual scenes, because the scene that is inside of our current field of view is typically only ever a subset of our broader panoramic environment. And we explore this environment actively through head turns and body movements, constantly exchanging what's in our current field of view with our memory of our surrounding environment, and using our memory to set up predictions about where we'll look next and what we can expect to see when we do. Yet, Traditional models of visual processing typically stop up at scene recognition, leaving out all that memory brings to bear on visual processing out of the picture. Today, I'm going to argue for a more comprehensive understanding of visual processing that really recognizes the intimate link between memory systems and visual systems, and studies these processes in the naturalistic context that I think tend to naturally and fully engage their interaction. So first, I want to emphasize that studying the link between perception and memory is a hard problem in cognitive neuroscience for a couple of reasons. The first is just about our paradigms. We don't typically have paradigms that really stress real-world perceptual mnemonic interactions. In contrast to our everyday experience, like I've just tried to illustrate here on the screen artificially, our traditional paradigms in psychology, usually involve humans that are head-fixed and looking at small images on computer screens. Those images are probably passively presented to the participant. And of course, these types of conditions are really necessary, really helpful when our goal is to carefully isolate perceptual versus mnemonic processes. But they often eliminate some of the natural opportunity for perceptual mnemonic interaction that's characteristic of active vision. So second, on the neural side of this issue, memory and visual systems have historically been studied in isolation in our field, with rather little integration between these literatures. Then the characteristics of these systems are often thought of as kind of opposite in nature in terms of the representations that they're thought to compute, so internal models of the world versus processing external stimuli, their temporal orientation towards the past versus the present, the reference frames that they're thought to use, allocentric versus egocentric, and the properties of neural tuning. So it's really unclear how these two systems could interact even in principle, just given that they don't speak a common language. Current models of perceptual mnemonic interactions really don't answer this question too thoroughly. So in the classic, say, Van Essen view, you might probably picture a network like this. And we think of visual and mnemonic structures as situated on opposite poles of a visual hierarchy. And each stage along this hierarchy probably marks some type of transformational step from the language of vision towards the language of memory and back. But this model, of course, leaves a lot of ambiguity about what those internal transformations, what those transformational steps are, what they look like, as well as where along the visual hierarchy our memory first starts to penetrate our visual processing. These are some of the deepest questions that I think we contend with in cognitive science in particular. But this knowledge gap also represents an important gap from the clinical side, because this interface between memory and perception seems to mark a fault line for multiple different psychiatric conditions. So for example, in autism, 90% of people report differences in low-level sensory processing, including vision, and these differences are known to scale with cognitive level symptoms, including in memory. But the link between these sensory and cognitive symptom domains is not well understood. And similarly, probably more famously in dementia, a significant risk factor for memory decline is actually sensory impairment, both in the auditory system and in the visual system. And it's even the case that the degree of sensory loss predicts memory loss, as I believe I'm showing here in this figure. But little is known about the biological underpinnings of this association. So understanding how memory and perceptual systems interface is an important question, both from a theoretical and a clinical standpoint. And this is the big question that my lab is interested in. How do our perceptual and mnemonic systems interface in the brain and behavior? So my talk today is going to focus on 3 vignettes from our lab that have been exploring this overarching question, primarily from the neuroimaging point of view. So first, we're going to ask how perceptual mnemonic systems interface in the brain. I'm going to focus in particular on some insights from around high-level visual cortex, introducing you to a new functional motif that we think is important for enabling memory and perception to interface during scene understanding. Second, We're going to explore some literature in which we've been asking what coding principles within these areas of the brain are enabling memory and perceptual systems to interact despite their different native languages. And finally, we'll ask how memory, what role even the apex of memory systems, the hippocampus, might be playing in structuring perceptual mnemonic interactions. I just want to highlight that all of these different areas of work in my lab were run by these amazing people. I'll especially note my former postdoc, Adam Steele, my current postdoc, Peter Angeli, and my students, Anna and Brenda, and my collaborator, Ed Silson. Okay, so we'll start here with the first question. So when we think about memory signals in high-level visual cortex, I'm circling somewhere in this network here and saying high-level visual cortex. The first concept that comes to mind is probably the idea of reinstatement, which holds that the same regions of the brain that initially encode a stimulus also reactivate when you start to participate in top-down recall of that same class of stimulus. So here I'm showing you the scene selective regions of high-level visual cortex, PPA there on the ventral surface and OPA here on the lateral surface. And this view, of course, is deeply rooted in studies from the mental imagery literature that show that category selective regions like FFA on the top here or PPA on the bottom here are not only selectively active when people see their preferred stimuli, so a face versus a house versus a face versus a house, but also when they simply imagine their preferred stimulus. So many studies have replicated this effect, leading to the widespread view that high-level visual areas of the brain support both perception and visual recall through reinstatement. But reinstatement presents a problem from the perspective of theoretical neuroscience. How do memory and perception coexist in the same neural population without causing interference? So unlike these fMRI studies, which typically probe memory and perception serially, our naturalistic perception requires that we simultaneously represent, say, the scene in front of us, while maintaining a memory of the surrounding environment to predict what we might see next as we move around. But if the same neural populations are concurrently used for both perception and memory, as reinstatement suggests, of course, this would cause some level of interference. So how does the brain reconcile and prevent this type of interference? We think that resolving this tension represents a big challenge right now for understanding how perception and memory interact in neuroscience today. And recent studies from our lab have made some headway in starting to understand this perceptual mnemonic interface in the case of scenes. So in short, we found that the classic reinstatement story doesn't appear to hold for scene selective cortex. So if we carefully localize scene selective cortex using a visual localizer, like the one I'm showing here on the right, where participants perceive pictures of scenes, or faces, or bodies, or objects, and we isolate those classic scene perception areas of the brain, PPA here on the ventral surface, OPA here on the lateral surface, as regions that preferentially activate during the blocks of scenes as compared to other classes of stimuli. We isolate those areas, OPA and PPA. But when we change their localizer paradigm to focus instead on scene memory, so now we're asking participants to remember scenes versus other categories. So remember your kitchen at home versus the face of your mother. Something striking happens. We find that scene selective memory activity shifts immediately anterior and adjacent to those scene perception areas, both on the ventral and the lateral surface. So I'm showing you this illustration here in the case of one participant's brain. But I want to emphasize that that anterior shift in our initial study was consistent across every participant in that study. And I've collapsed that data here just to show you in the case of Rose plots, each person here is a vector. And the origin of that Rose plot is the center of mass of their scene perception area. So on the lateral surface, the center of mass is their OPA, ventral surface, their PPA. The length of that vector is showing you the length of their anterior shift for memory as compared to perception. So interestingly, this shift is not present in our hands for other category selective areas of the brain, like FFA, suggesting that this effect might be specific for scenes. So since publishing that original paper, we've actually replicated this anterior shift for scenes, for place memory relative to scene perception, in 2 additional studies using various different kinds of localizer approaches, dynamic stimuli and static stimuli, different types of memory tasks. And each time we've observed the same anterior shift in all of our participants. So for the sake of handles throughout this talk, I'm going to refer to these areas now instead of calling them by their names like PPA and OPA. I'm going to refer to the place memory areas as PMAs and the scene perception areas as SPAs. They'll either be on the lateral surface of the brain or the ventral surface of the brain. And additionally, I wanted to highlight that work from other groups, including Michael Rugg's lab at UT Dallas, has replicated this anterior shift in their own paradigms and even shown that it is amplified by aging. So the degree of this anterior shift, in this case, I believe we're looking at their lateral surface, is stronger for individuals who are older than individuals who are younger, hinting at potential clinical relevance for memory. So this is a good interim summary point. When we are thinking about this anterior shift for place memory versus scene perception as a functional motif in high-level visual cortex, it's replicated both on the ventral and the lateral surface of the brain. And it might be optimized to resolve this challenge of interaction without interference, positioning perceptual and mnemonic areas close to one another while maintaining distinct neural populations. This might be a good point for me to ask if there are any questions before I go on and show you a little bit more about the anatomy of these areas and their connectivity, as well as their own respective functional purposes and different tasks. Any questions here?
I will be glad. Yeah, I'll be glad to ask. Hi, Caroline.
Hi.
So do you present the same stimuli that you are asking them to recall? Are you showing them their own kitchens, their own family members?
In our first studies, no. In our first studies, we asked them to recall their own family members, their own kitchens. So they provide a personalized list of things that they can vividly recall from their life. And then we contrast that with just a typical category localizer, so stimuli that everybody would see. So there's a big difference in our first studies between the degree of personal familiarity. But in a study I'll go on to show you soon, we actually trained people to learn the same immersive environments in VR, and they either perceive those or they remember those. And we replicate the same shift, suggesting that that element of personal memory is not what is driving this difference between the SPAs and the PMAs.
Okay, so it's memory, it's personal memory, but also different level of categorization also, right? Like
yeah, right.
Something which are more individuating versus higher level of the category. But
Yeah.
in your next study, that's not going to be the case anyway.
Yeah, well, you keep your eye open for that and tell me if you think we've addressed that in the next study. But you're exactly where we were at the end of the study. We wondered, what are the various things that are different in the task between remembering your mother's kitchen and seeing a picture of a kitchen? And can we sort of take apart each layer of those differences and isolate what it is that's driving this anterior shift? Okay, so maybe I'll go on and then there'll be more points for questions along the way. So I just want to take you into the anatomy of where these regions lie for the sake of arguing that these place memory areas seem to be pretty well positioned pretty well situated as an architectural bridge between our visual system and memory systems, especially the hippocampus. So first, you might want to note where they're located relative to classic retinotopic maps and visual cortex. You can see that both the area on the ventral surface and the lateral surface are positioned just anterior to, for example, parahippocampal cortex maps. They're on the ventral surface, suggesting that they're not strictly visual in nature. They're not exactly well categorized by our retinotopic areas. I'll say more on that in the next section of the talk. But they preferentially co-fluctuate with the hippocampus during natural movie watching. So during paradigms where we just have people watch videos of scenes, some of them are familiar to them because they're parts of the Dartmouth campus, others are not. And over that naturalistic time course of a movie, we just ask how much different areas of this network are correlating with the hippocampus as compared to V1 as proxies for the apex and the origin of that perceptual mnemonic hierarchy. This is what we see. So the scene perception areas, I'll call again from here on in the talk the SPAs, are not correlated with the hippocampus during naturalistic movie watching, but the PMAs are strongly correlated with the hippocampus. And of course, there's a dissociation there, where the scene perception areas, like PPA, are strongly co-fluctuating with the visual system, and the PMAs are not. So in this sense, we think of the place memory areas, again, as well poised to bring information from memory to inform scene perception in high-level visual cortex. More broadly, this is a little sketch of our current working model of this anterior shift. So we're thinking here again of regions of the memory system that might possess your memory of the broader visuospatial environment, and the PMAs as well poised to convey some of that information from memory to inform ongoing scene perception. But stepping back into the broader psychology literature, there are a number of alternative accounts of what might cause an anterior shift pattern like this, some of which Galit has already brought up, and we wanted to consider as well. So let me outline a few of them. So one account comes from early work from the 1990s by Alex Martin's group, which I think this body of work is really beautiful. And it suggested that naming the color of a black and white object activates a region that is slightly anterior to the proximate location of color processing areas like V4. So by this account, that anterior shift might reflect a transition from processing the perceptual features of a stimulus, like color, to representing semantic information that's associated with that stimulus, like the color's name. Maybe something like that, semantic knowledge, is going on in our case as well. A second explanation comes from a recent paper from Jack Gallant's lab, which showed an anterior shift for computational language model fits to neural responses that are recorded while people are just listening to stories as compared to watching movies. So their argument was that this shift represents a transformation from sensory representations of a story to more linguistic or multisensory representations of that same content. So far, it's really not possible in our series of studies to know which of these kinds of accounts could best explain our effects. Our personally familiar places that people have been remembering are probably full of their semantic knowledge that's associated with those places, maybe even multisensory memories of the smell of your mother's kitchen and such. And any of these different associations might be driving that anterior shift from the scene perception areas. So what we wanted to do next is try to strip away all of those different possible associations that come from personal memories and deliver to our participants the same structured, immersive environmental memories. So what we did is we situated them in head-mounted VR, where all the participants will study the same set of 18 different environments. And these environments are just photospheres of different places in the world that they've never been to before. So we've taken away any type of personal memory from this task. And later we'll ask people to either perceive or remember those stimuli and see if memory is driving those anterior areas, even for these non-personally familiar stimuli. The second thing we're able to do in VR is start to build up the amount of spatial knowledge that they've associated with that stimulus that they studied, so that we can test our idea that anterior response is driven in part by the degree of visuospatial memory you have of the environment. So in this study, what we did is teach people 18 different scenes across 3 different study conditions, and each condition parametrically increases the amount of visuospatial information they have to learn about the scene. So in the first condition, we call this the image condition. Participants just study one single view of an immersive environment. It's just a little sliver. Everything around them in that environment otherwise is occluded. So they can turn their heads, they can look all around them, but the only content that they can see is their initial field of view. In the second condition, participants studied that same initial field of view of an environment, but it's broadened with 270 degrees of panoramic context around it. And in the third condition, participants studied a single field of view, but it's broadened with a whole 360-degree panorama, as well as the ability to walk a little bit down that street. So the thing to take home here is that across these three conditions, a single initial scene view has been associated with increasingly larger amounts of spatial context. And next, we brought participants to the scanner and asked them to simply just recall that initial scene view from each of the studied places. We predicted that if those place memory areas reflect the extent of visuospatial memory that was associated with the scene, response in those anterior areas should scale across those three study conditions. And that is exactly what we found. So I'm showing you here activity in the lateral place memory area. And in contrast to that area, the PMA there on the lateral surface, you can see that the scene perception area shows no significant modulation of visuospatial context. And then to Galit's question in particular, you might even just compare the first condition here of the image condition. This is the condition where somebody is just imagining the initial scene field of view. And we're looking at how activity is higher in the PMA as compared to the SPA. And in the study, we have a flipped contrast where you're just perceiving that initial field of view, and you can imagine a scene perception area is driven above baseline when you're perceiving a scene. And I'll say for the sake of time, I'm just showing you here that one figure from this paper, so check out the others, and I'm only showing you the lateral surface, but the same pattern is also observed on the ventral surface for the PPA and ventral place memory area there. So importantly, the place memory areas are showing a strong response in all of those conditions. But I also wanted to just add at this point that it's not just that these regions are activated by memory in this case, but also that we can decode the particular environment that the person is remembering in those particular trials. So taken together, we think of this as evidence that those PMAs may in fact be processing the memory of the broader visuospatial content that's associated with a scene view. And before wrapping up this study, there's just one more feature I want to draw your attention to in this data. It kind of kicked off the next two sections of my talk. So you may have noticed that that scene perception area, OPA, isn't even responding above baseline in any of those recall conditions. And in fact, as you can see from some of the subjects here in gray, I don't know if these are coming across nicely on your screen, but some of our participants even have response that is suppressed below baseline during this visual scene recall task. So here I'm going to just pull out the time course of their response from that same set of data. And this is kind of nice because you can look to see how the activity in the PMAs is rising over time after the cue to recall. The activity in the SPAs is actually going below baseline around the time that the PMAs are peaking. So we looked at this time course and we thought, wow, that looks like a little bit of a push-pull effect there. It's almost as if as you start to initiate recall, you're suppressing the scene perception areas. We weren't sure at this point what this type of effect might be for, but one idea is that an effect like this is often consistent with models of predictive coding, where higher order brain areas suppress lower level sensory areas as they deliver or convey a prediction about upcoming sensory input. So this idea has kicked off a number of studies in the lab trying to explicitly test whether this functional motif might constitute a hierarchical predictive coding circuit for scene processing, where the place memory areas are providing information from memory to facilitate ongoing perception. This is another great point to here, I'll summarize first and then take a question. So all in all, the data so far are showing a clear anterior shift for place memory relative to scene perception at that anterior edge of high-level visual cortex. This anterior shift seems to represent a segregation of mnemonic and perceptual information associated with the scene. These place memory areas are co-fluctuating together as a network and also interfacing with the hippocampus. And we know that they represent specific remembered environments. From here, our interests are starting to understand whether, like, basically just what this functional motif is for, what's it doing, and how do how does this motif, which seems to currently just suggest segregation or separation for memory versus perception, how does it set us up now to interact across the perceptual mnemonic divide? That's what the next two sections of the talk will be about. But this could be a good point for just a question about the motif itself.
Caroline, I'm not sure this is maybe I leave it till the end, but I'm just wondering whether your dorsal PMA, if I remember correctly, then name is in any way overlapping the DMN, the default mode network area, which is quite close, at least anatomically, to where it is highlighted here on the
Yes.
right. So, but you don't have to answer that. You can,
Well, this will be actually very relevant for the next section of the talk. So I'm just going to preview the answer because you've asked it so clearly, and I have a figure that very clearly answers it. But the
I
next section
see.
of the talk is going to be about stepping into the default mode network and understanding the interaction between that classic large-scale network that's associated with memory and internal retrieval processes and the place memory areas. So
Okay.
you can see here the sort of pattern on the lateral surface. If you were to move on average from where a parcel would locate OPA in white to
Mm-hmm.
where our parcels would locate the PMAs in yellow, sorry, in orange,
Yeah.
um,
Mm-hmm.
you sort of step right into the default mode network. And we do overlap with that key node of the default mode network there.
Wow. Nice. Very nice.
Very weird.
Yeah. Very nice. Very interesting.
Okay,
Thank you.
so
Can I
here
ask
we
a quick question? Maybe
yeah,
you'll answer it later. So there is also an overlap between the perception and memory area, right? I mean,
yes,
there's the anterior shift, but there's also an overlap. Did you look specifically at the overlap? And
yes.
maybe this is part of the interaction you're going to talk about later.
Yeah. So what Galit is pointing out is, even in this figure here, I'll focus on the lateral surface because I feel like it looks like a Venn diagram to me. But you can see that OPA, a good section of it does not overlap with the PMAs, but some of it does. And similarly, the PMAs overlap with, the keynote of the default mode network there on the lateral surface. So are there any functional differences in responses as you move from like pure scene perception area into multiplexed scene perception and place memory cortex? Honestly, at this point, I don't know the answer to it. So in our studies so far, everything that we do is either analyze our responses along a continuum across, or we isolate, we take the top 200 voxels that are the most scene selective and compare them to the top 200 voxels that are the most scene memory selective. And those tend to not be in the overlap zone. So we don't know whether there's something, some type of different transformational representation living in between those two areas. But there might be. Yeah. Okay. So I'll pull us back to where we were in the talk here, and we're going to move now. So thank you for setting up the pivot into the default mode network, because if you're like me, I really was a vision scientist to start. I never thought about the default mode network. I did not want to think about the default mode network. And it has entered my life. And so here we go. We're gonna be in our place memory and scene perception areas to start in this section of the talk. And then we are going to find ourselves in these large scale distributed memory networks in the brain. Okay, so this section of the talk is about the voxel level coding schemes that seem to inhabit these more mnemonic versus more perceptual areas, to try to understand what kinds of coding schemes might structure the communication across these areas. So I'll step back and just refresh our memory for why this might be a puzzle. Of course, when we think about something like retinotopy, by which I'm gonna mean here simply that a voxel has a preferred location in the visual field in which it responds to a stimulus. So I'm not necessarily referring to the cortical layout of voxels that respect the layout of the retina. But of course, we know that when we look at either the ventral or the lateral surface of the brain, you see a series of retinotopic maps. So this is the topographic layout of the retina as you move from view one, stretching anterior towards these regions that we've been interested in. And indeed, the classic view is that those maps, of course, are robust and there on both surfaces of the brain, but they eventually stop. The classic view is that retinotopy eventually is shed as a coding principle as information propagates up the cortical hierarchy. And this is because we think of memory regions at the cortical apex, like the default mode network, as needing coding schemes that would support internal and context-dependent information processing, rather than a neural code that respects the way our light hits our retina. But if retinotopy is lost in memory structures at the cortical apex, what neural coding principles are enabling perceptual and mnemonic areas to interact? So how do visual and memory areas interface without a common neural code? Well, a pattern has emerged recently in the neuroimaging literature that we think might provide a partial answer to this question and also challenges our traditional view of the cortical hierarchy. So basically, it's become clear that much more of the brain has a retinotopic code than was previously thought. So here's a figure from one of our recent studies, though I want to highlight that this initial observation was made by Thomas Knappin's group in the Netherlands. And what multiple studies now have observed is that when you use a population receptive field model to identify which voxels in cortex respond when a stimulus is located only in one rather than other parts of the visual field. So which have a visual field location that they preferentially monitor. Every colored voxel on this image it is an area of the brain that has what I will refer to here in the talk as retinotopy, a retinotopic preference for a specific visual field location. We refer to this as a receptive field, of course, or a population level receptive field in our case, because our spatial resolution in fMRI is at the level of a population of neurons. And I'm going to call this type of preference for a visual field location the voxels PRF throughout the talk. Every voxel in this talk is going to have a particular center position to that PRF, a particular location in the visual field that it monitors. I'm being a little cagey when I say I'll call this retinotopy because, of course, we know as vision scientists that retinotopy is often a word that is reserved for talking about that cortical topography that respects the layout of the retina, where neurons near to each other represent the same nearby parts of the visual field. But as you'll see as we go on here, we end up with much more of a salt and pepper shaker neural code as we move up the cortical hierarchy, where voxels have particular locations of the visual field that they monitor, but that doesn't necessarily mean that they're next to ones that monitor the same location on the cortical surface. So you'll see as this goes on. Okay, so these results alone, before I get into the color scheme of this map, already highlight the surprising prevalence of the retinotopic code throughout the brain. But what's even more surprising here is the emergence of these cool colors. These are regions of the brain that have a negative retinotopic code. So while voxels in visual areas show a classic positive response to a visual stimulus where their activity rises when a stimulus is presented in their preferred visual field location, a significant proportion of the brain actually shows what we call a negative retinotopic code, meaning that voxels in these locations tend to be specifically deactivated when a visual stimulus is presented in their preferred location of the visual field. So just to give you an example of what this looks like or a better intuition of their time courses, these are two voxels. The top comes from a default mode area. The bottom comes from a visual area. Actually, I could also say the top comes from a PMA and the bottom comes from an SPA on the lateral surface. And these, I've picked voxels whose visual field location preferences are pretty well matched. So the top one likes the bottom right quadrant of the visual field and the bottom one likes a pretty similar portion of that quadrant. And you can see that whenever a stimulus passes by its preferred visual PRF location, the bottom one is activating above baseline, but the top one is deactivating above baseline at those same moments. Okay. So this negative
Um,
retina, yes,
could,
good time
what's
for
the
questions.
scale on what's one plus and one minus on that thing in degrees of visual angle or something?
Oh, sorry. Yes. So that it just says on the axes of the visual field, -1 to 1, because with the model, you tell it the size of your actual visual angle, that you're displaying in the scanner, and then the output it's normalized to that size of the visual field. So it's normalized to just -1 to 1. In our case, it depends on our study, which scanner we've used, how large the visual field is, right? So in, in this study, uh, visual angle in that scanner, I think would have been, I don't want to get it wrong, Jeremy. I think
I'm just
maybe
trying to
about,
get a feeling for whether or not those spots are lying near the fovea or somewhere out in the periphery.
so imagine you're in the scanner and this is your visual field. It's about this much. It's like the size of the mirror that you're looking up at in the scanner. And sometimes we've had, a larger mirror than other scanners. So I'm not exactly sure what the exact visual angle is, but these spots are closer to the fovea, which is in the middle of that screen. And then out towards the periphery would be, 15 degrees.
Okay.
So we are not working with a far periphery for sure. We're barely working with a periphery. Um, any other questions here? I'll say just as a point of interest, that's one reason actually that we see some negative, responses in peripheral visual cortex there, if you can see my mouse. There is a little bit of a surround suppression effect and peripheral cortex that is unrelated to the memory effects we're going to talk about next. Okay, so we became interested in this question a few years ago, given our interest in coding principles that the brain might be using to structure perceptual mnemonic interactions. And specifically, we started to wonder if this negative PRF at the cortical apex, this negative PRF population there, might represent a latent visual code in memory areas that might be helpful for structuring interactions with visual cortex. So the first thing we did to start to explore that idea is turn back to those place memory areas and scene perception areas, because we noticed that they straddled the positive to negative transition on the lateral surface. And indeed, if we just quantify the proportion of negative PRFs in those two areas, you see that in OPA, the lateral surface's scene perception area, there are barely any negative PRFs. Whereas in the lateral place memory area, about 60% of the voxels there are negative as compared to positive PRFs. So the vast majority in OPA are positive, and the majority in the PMA are negative. So what that means is that when a stimulus is presented in a specific location of the visual field, a voxel in the scene perception area that monitors that portion of the visual field is driven positively, but a voxel in the place memory area is suppressed below baseline. So far, this just shows that the memory areas have a retinotopic code with a negative sign during visual stimulation. I'm going to say, for the sake of time, in this study, when we flip the question and have people recall a stimulus in a location of the visual field, we find that that pattern flips, where now we can drive the place memory areas, which used to seem to be suppressed responses above baseline, and relatively suppress the PRFs and the OPA. Again, in a retinotopically specific way. So in other words, those sort of suppressive effects that I showed you earlier in the talk turn out not to be global, but are specific to voxels that monitor the location of the visual field that is being stimulated, that you're either doing memory in or you're presenting a stimulus in, compared to voxels that monitor different locations of the visual field. So that suggests that the retinotopic code is structuring an opponent interaction across these areas during visual and memory tasks. So we wondered, is this retinotopic coding scheme something that the place memory areas simply adopt when they're doing scene processing tasks? Or is this more of an intrinsic functional motif that is there even in their just natural spontaneous interactions during rest? So to ask this, we wanted to get a sense of the activity dynamics of these perceptual and mnemonic areas in task-free conditions when no stimulus is present. So we turned to another data set in which we were able to localize the place memory areas and the scene perception areas in people and perform retinotopy on them. So this is from the natural scenes data set. But then we also had resting state scans on the same individuals. So after identifying the retinotopic preferences of particular voxels in these areas, we simply asked whether perceptual and mnemonic voxels that monitored the same location of the visual field were anti-correlated at rest, something like that. So that voxels that monitored more so than voxels that monitor different locations of the visual field, say a blue in the top right quadrant and a red over here. And this is exactly what we found. So over the time course of a resting state run, this is an actual set of data over a resting state run that goes from 0 to 195 seconds there. And I'm showing you the time course of activity from a negative PRF in a place memory area in blue and a positive PRF that monitors the same location of the visual field in red. Again, these are in different areas, PMA, SPA, and you can see that those two time courses are co-fluctuating in antiphase with one another. Now, if you're aware of the default mode literature, you know that this is sort of a global effect that's been observed across large-scale default mode networks and dorsal attention networks in the brain, that we think of those networks as at rest and in antiphase with each other with spontaneous dynamics. But I want to modify that global understanding of those two networks here and say it is only true, that antiphase relationship is only true as a function of retinotopy. So if we take now a voxel that monitors a different location of the visual field, this is now the lighter red color here, you see that that antiphase relationship is reduced. And if we move from there out of our focus on the PMAs and SPAs, and instead, as Sharon was hoping to see, now define the entire default mode network in those people and compare it to the dorsal attention network in those people, we find the exact same effect, where if you take negative PRFs in the default mode and you take positive PRFs in the dorsal attention network, they are co-fluctuating in antiphase with one another more if they monitor the same if the voxels monitor the same location of the visual field compared to different locations of the visual field. So we think, all in all here, that not only is retinotopy present in mnemonic areas of the brain, but that this latent population level code in these memory areas might be useful for rotating the representations of visual stimuli into a different, along a different axis. In this case, it's a 180-degree flip, and structuring opponent dynamics across these large-scale systems. Okay, that's a good point for questions. Or I can just plow on and do the last few slides on the hippocampus and take it all together from here.
Yeah, that sounds great.
Okay. So the final section of my talk, we're interested in now moving into the hippocampus. This is all unpublished and new, needs feedback to understand how this bivalent retinotopic code might extend into what we think of as the deepest memory system of the brain. So again, contrary to the classic Van Essen view, there's actually good reason to think that the hippocampus might participate in visual coding. So first, of course, the hippocampus is strongly coupled to the default mode network, which we know contain a large proportion of negative retinotopic voxels. And in the DMN, we've shown that these voxels are driven by visual recall. So it's plausible that the hippocampus might employ a similar voxel-wise retinotopic code to coordinate hippocampal cortical interactions around visual memory, visual retrieval. This seems particularly plausible given recent work in fMRI in humans, showing that the hippocampus has strong visual field biases, where the left hippocampus preferentially responds to stimuli in the right visual field and vice versa. And also related work in macaques and non-human primates, showing robust visual field coding in neurons in the hippocampus, thinking of Julio Martinez Trejo's work in particular. So we wondered whether this bivalent retinotopic code that we've been following might extend all the way up the cortical hierarchy to the hippocampus. And this is basically the answer. So to test this, we looked at retinotopic model fits in the hippocampus in participants in the natural scenes data set. So these are 7T scans, which are nice and high resolution, which is important for getting into the hippocampus here. And what you can see is that in each of these participants in this study, there are only 8 of them in this study, all of them show a strong bivalent retinotopic code in the hippocampus. Importantly, if you take any one subject and you threshold that code to only significant voxels, I want to emphasize it's not that the entire hippocampus shows retinotopic preferences. Only about 10% of voxels in any participant in the hippocampus are well fit by a retinotopic model. So this voxel-wise code is only present in a very minority of hippocampal voxels. But that said, for those voxels, there are robust positive and negative responses. These are real-time courses here from our data. And they are retest reliable across runs, where voxels maintain a stable preference for a particular center position in the visual field across different runs of this scan, even in odd runs, as well as a consistent preference to be either a positive or a negative PRF. So again, if you just want to look up the visual hierarchy, moving from early visual cortex to PPA, the PMA here, and the hippocampus, we see that the vast majority of voxels and visual areas are showing a retinotopic response, but a small minority in hippocampus are like this. That said, if you look at the proportion of voxels that are negative as compared to positive, that proportion increases as you move up the hierarchy towards memory areas. Finally, these negative PRFs in the hippocampus, I'm just going to make this statement and then kind of wrap up here for questions, appear to coordinate functional coupling to cortex. So to test this, we turned again to the resting state data, where we select voxels from the hippocampus that are negative PRFs during retinotopy, and then we find that they are preferentially coupled to positive PRFs in cortex at rest in an oppositional way, where if 2 voxels, either in the hippocampus and cortex, monitor matched regions of visual space as compared to different regions of visual space, they are more in antiphase relationship to each other during rest. In other words, hippocampal cortical connectivity at rest is scaffolded by what we call retinotopic alignment, a shared visual field bias between 2 voxels, and those voxels living in the hippocampus and out in cortex. So where in cortex is coordinating with the hippocampus by this retinotopic code? If we take all of the different large-scale cortical networks in cortex that we've defined individually in our participants at rest, and break them down into, for example, the default mode network over here on the left, we find that this effect is primarily driven by hippocampal cortical connectivity to the default mode network, where the hippocampal PRFs are functionally coupled to DN PRFs as a function of their retinotopic alignment, their shared visual field preferences. So far, this set of data is telling us that about 10% of hippocampal voxels are retinotopic, as in being well fit by a PRF model. This retinotopic code structures hippocampal cortical interactions at rest, particularly with the default mode network. And we wonder now whether this bivalent code might be somehow useful structurally for coordinating memory versus retrieval states, encoding versus retrieval states, that should say. So top down, bottom up between the hippocampus and cortex. So zooming out, the big claims I think this body of work is trying to make right now are 3, and I'd like to try them out on you. So I've told you today that naturalistic visual experience involves an interplay between our memory and perception. And I've introduced you to a set of studies that are shedding light on the functional encoding principles in the brain that we're using to interface perceptual mnemonic systems. We think these studies are showing that scene memory relies on a dedicated functional motif at the anterior edge of high-level visual cortex. We think of these place memory areas as forming an architectural bridge between high-level visual cortex and the hippocampus, possibly serving memory-based scene prediction and perception. We think that perception and memory share much more in common with their neural codes than were previously presumed. So across the brain, voxel-wise opponency between positive and negative population receptive fields create a push-pull dynamic across perceptual and mnemonic areas, possibly enabling interaction across these systems while preventing interference. And finally, the hippocampus and the default mode are embedded within this visuospatial scaffold. So functional coupling across these large-scale networks, like the DMN and DATN, or the hippocampus and cortex itself, seem to depend on retinotopic features, which I think prompts us to rethink models of the cortical hierarchy that keep and confine our visual codes to the visual cortex. So taken together, these studies emphasize the intimate link between memory and perception and gesture towards one of many frameworks, underlying frameworks, for understanding how the brain is weaving together our internal and external representations of the world into our coherent experience. So thank you guys. Excited for more questions. And I also, again, want to just especially thank the key individuals in my lab who have driven this work that are named up here at the top.
Thank you so much, Caroline, for a really great work and exciting presentation. And I want to invite everybody to unmute yourself and give Caroline a big applause for a great, really a great work. And if it's okay, I mean, you just said you're happy to take questions. Okay, you're getting, oh my God, that's so nice. I think that's a new Zoom feature. Hand claps. Great. I want to open the floor for questions. I myself have a question. I mean, because so I'll start. You said that there's some kind of like a push-pull, and you're looking at scenes, and for example, that are associated with peripheral or let's say, whole visual field representation, whereas, for example, faces or memory of faces is more, let's say, assumed to be focusing on foveal. And do you think that would match, for example, more foveal? So you would see foveal biases in regions that may code face representations or memory of face representations? Or, I mean, do you think this would generalize to additional categories? That's one question. And another question I have about the 7T, I was wondering because you were looking at, you showed single subject maps, and I wasn't sure whether the spatial arrangement between the positive and the negative was similar across participants. And I wondered if this is some kind of like, so would you find that this is present in each one of the participants, but the topographical or arrangement within participants, or is that similar too, or do you find it some individual arrangements within the hippocampus? But you don't
Within
have.
the hippocampus, okay.
Yeah. So you showed the actual individual maps of the more or less the red and the blue, and I was wondering. I mean, I didn't see.
Yes.
Maybe I missed it, but it didn't look like a really consistent
Okay.
pattern of red and blue voxels. So I'm just wondering.
So these are all great questions. I tried to jot them down, but ping my memory if I forget as I try to back through them. I'm going to go. I'll go in reverse order. So regarding the spatial arrangement of positive and negative voxels in the hippocampus.
Yeah.
So as you noticed here, we're showing you unthresholded data here where it's less clear like which of these voxels end up becoming significant PRFs. But you don't see any obvious topographic distribution of PRFs along, say, the AP axis in the hippocampus in this participant. And that is correct even at the group level. There is no, in our data, no obvious pattern of whether, say, AP or particular subfields have more or less PRFs generally. Now, regarding the amplitude of those PRFs, are they negative or positive, and whether there's something consistent within participants about that, I consider this like a salt and pepper shaker, like distribution of that code. Basically, no. There is no difference in the proportion of negatives along any axes in the hippocampus or different subfields. I wonder if I actually have an example of that, but maybe just for the sake of time, you can take my
I
word for
completely
that.
trust your your answer.
And so then I'll jump to the theory of why we think that is. This is all still, to be tested, but our idea here is that the hippocampus, kind of like we find in the place cell literature in the hippocampus, might actually make use of a code that is not confined to the retinal like the retinal topographic arrangement that visual cortex has. So you might imagine that in a memory system where my goal is to start to bind together associations about something that's in this part of my visual field and something that's in a different part of my visual field to build a coherent understanding of a scene, it would be to my advantage actually cortically or within the population topography in the hippocampus, not necessarily to respect the layout of the visual field, but to allow for neurons to be near to each other that grab information from different locations in the visual field. So similarly, in the place cell literature, you actually see that, where the place cells are distributed in the hippocampus when you enter a new environment, actually it's kind of random which place cells become place cells in a new environment. And also there's no, near neighbor distribution where like a place cell that represents this part of an environment is right next to a place cell that represents a different part of the environment in the hippocampus. So we actually think that part of the rule of moving from perceptual to mnemonic systems might be breaking the interest in respecting the retinal layout. And so within participants, there's no particular pattern either.
Okay, great.
So that was one question that I'm really excited about. This hippocampus work is very new, and we're excited about sort of testing, that theory there. But it's very much a theory. Single subjects, okay, foveal biases, different categories. Different categories in our hands do not show the anterior shift. I showed you the example of faces, but right now in my lab, there's a wonderful student named Deepa Prasad who has rerun the study with many, many, many different visual categories. And do I have one of her figures here? Not sure. But she shows that whether you're imagining faces or bodies or objects or scenes, and whether you look at OFA, FFA, even anterior face areas, the only region in our hands that shows this anterior shift are the scene selective areas of cortex. So why? Could it have to do with the foveal peripheral biases? Is that where you were going with that next part of the question?
Yeah, I thought, I mean, I thought, but maybe, but
Yeah.
that's a great, that's really interesting. I mean, it's even more fascinating if it's only found for the scene selective regions. So
maybe there have been times where I wanted it to be found for others because some earlier ideas, of mine, I did think it might appear for other categories as well. So we're still very much on the journey of interpreting this.
Thank you.
Maybe, I didn't connect back to the foveal and peripheral biases, but feel free to bring that back up if you'd like.
Yeah, and I'll be in contact with you about more questions that I have, but I see that there are so Jeremy and also Jody. Are you okay to take questions?
Yes.
Okay, so Jeremy, do you wanna unmute? You are.
Yeah, so to pick up on really what you were just talking about as who and who does not show this anterior shift. As a visual search guy, my interest might be more in objects, and I might assert that the problem that you started out with of how do you keep memory and visual stimuli straight is perhaps more acute at the level of being within a scene and wanting to keep track of objects. If I'm in the kitchen and I get a fork and then I want to find where the salt is, but I've turned my head, and so the salt that was in my visual field is now out of my visual field, but I'm thinking about the salt. I don't want to confuse my memory for the real object. So it's very much the same kind of problem, except that I would argue that it's happening all the time, rather frequently. So how are you going to solve that one?
I thought we were agreeing there. So I think the first thing you said is that among classes of stimuli, this problem of having the requirement to maintain in working memory what is outside of your field of view is stronger in the case of scenes than other classes of stimuli, unless they are embedded in scenes. So for example, if I look at an object like my cup, I don't really have a strong requirement to keep the memory of the backside of the cup in mind as I process it and recognize it as a cup. I do have a strong memory to remember where it is when I'm cooking in the kitchen if it's outside of my field of view and embedded in the broader scene. So I guess my top level question is to answer to why do we think scenes are different? Is I think that compared to every other category that we study in the visual system, they don't fit into one single field of view. And also the initial requirement of interacting with an object or a face rarely requires that you remember the back of the head and such quite as much as with a scene, how I need to keep in mind what's outside of my field of view to be able to turn and reach and get something inside of it.
Right. I think I would totally agree that the keeping track of the back of the head or the back of the cup is not interesting. It's keeping track of specific objects within a scene context.
Yeah.
Right. That if as you say, if you put that cup down, you really want to know where it is to pick it up again.
Yeah.
But you need to keep that version of the cup different than the visual version of the cup.
Because you've transformed it in that case, like the relevant properties are where it is rather than like it was at the blue cup, for example. Is that what you mean, that version of the cup compared to the perceptual version?
Well, I mean, you could point to the cup in both cases. In one case, it's in the visual field, and the other, in your particular example, I think you're pointing over your back shoulder. But you could do it, and
yeah.
Are those somehow a similar representation of the cup within a scene representation? Or is that a cup representation that you could watch going in and out of memory in some fashion?
Going in and out of memory. Interesting. Yeah, I mean, that's a very good question. So I guess another way to rephrase it is to say, if we're saying that right now, when you isolate visual categories, the only category that seems to show this anterior shift is scenes. Cool. But now what if you situate objects within scenes, or you situate faces within scenes? Would you see an anterior shift for face perception or object perception versus memory if you're in this context where the stimulus is moving into and out of your field of view and you're required to keep it sometimes in working memory versus process it in your visual field? And these are great questions that we have not yet answered, but would love to.
I yield to the Jody from the north.
Thanks, Carolyn. That was an awesome talk. Very elegant work. I think you largely answered my first question. So there's these the negative population receptive fields, including in the more lateral parietal bits. There's no topography to them. They're just kind of higgledy-piggledy, if I understand. So you don't see any topography, even like, say, upper lower visual field or central peripheral. Is that the right understanding?
No. So just like how I showed very clearly that the hippocampus has visual field biases, left to right, So similarly, we do see both upper, lower, and left, right visual field biases to the lateral versus ventral place memory areas, just as you do for the lateral versus ventral scene perception areas, and the right versus left, right? But then if you look within one of these regions and ask, is there any cortical topography? So this is an example individual here from a slide that I didn't show you. But is there any cortical topography within that person's LMPA that shows something like a map? That we have not seen.
Okay.
So yes, visual field biases are respected just as they are in the visual system, to the level of quadrant.
And your stimuli in this case, these were just flickering checkerboards. I'm thinking back to some of the old stuff Marty Serino had done with Buffy the Vampire Slayer Toppy, where they'd had more complicated stimuli.
Yes. Yes, we also use more complicated stimuli, and we believe that that is important to driving the most signal in these higher order areas. So if I have a picture of the stimuli, they are just natural scenes that are shown within apertures. But I apparently can no longer click on my...
Okay. Last question. So you were working with 7T. I don't know what kind of spatial resolution you had, but have you done anything with layer-specific activation to look at bottom up versus top down interactions between these higher...
Not yet, but that would be a really awesome direction. Yeah, not yet.
Okay, thank you.
So just a quick
Yeah.
question. So I'm thinking about this idea of like the larger visual field in the scenes that activate more the memory areas. If you were asking people to recall faces and objects from non-canonical views, so they have to kind of like generate these spatial, I don't know, represent images, right? I'm just trying to think what would parallel this kind of like large scale spatial memory, right? Which we probably have naturally for scenes, but not for objects and faces. So whether that would be, I don't know.
Well, you guys tell me what you think about this claim, but like I think that to some degree, everyone is focused, including myself, on like why scenes and not other categories. In part because we think of scenes, faces, bodies, objects as all high-level visual categories of the same class or something. And I think that's partly just a historical accident in the field where, the FFA was discovered in the 90s and the PPA shortly after that, and we sort of put them around the same location of the visual hierarchy in our brains. But I'm just not actually sure that, I'm just not actually sure that we need to think of scenes at the same level as these other visual categories. And so if we break that assumption, then the question becomes more like, the scene is the container in which all of these visual categories in a natural world like appear. And what are the kinds of paradigms where you could situate faces and objects and such in scenes that would elicit this type of this requirement to have this, close relationship between what you have in memory and what you have in your current visual field. And then, what region of the brain would show that distinction between the memory and the perception? And it might very well be that it's still what we call the scene selective, like this functional motif for scenes, that is the place where you would decode the face that's out of sight, or the face in a non-canonical view, or the face that's in your periphery, like whatever it is that's going to be what drives this holding the representation away from the current field of use representation. It might be in what we think is scene selective cortex rather than it's like the FFA itself that shows an anterior shift, if that makes sense.
Mm-hmm. Yeah, I guess if the question is a general question about perception and memory, then we want to know also what's the case for the other categories with respect to perception and memory, right? And the different codes, similar codes, how they translate. It's a general question. So how it but
still TBD. I think what are the paradigms that pull out that same requirement for other categories, and then where would you observe that in the brain?
I have to add here, although I wasn't asked about this, but I see scenes as scenes are always there. Like the fact that we are living and present, we've got a scene. Once we may not have objects, we may not have people, but the scene will always be there if we are there and we open our eyes. So that's like a prerequisite of our vision. I mean, I'm not talking about all these, commercial things where they eliminate a scene and it looks like something is just present and midair, but a scene is the basis. And maybe that's why it is different than other categories that we may have different. Objects. So Jody's here. We have,
Yeah.
it's not only where they are or where we should look for them. If we're talking about Jeremy, we have, the representations are more complex because actions come into play and it's more complicated. It's different type of we don't move a scene usually. Well, maybe furniture, but the scene is just so maybe that's just yeah, another
Well,
thought.
Yeah.
are they really that different though? If I say, oh, let me go from here to the bathroom. Am I pulling out a bathroom object in my brain or a bathroom scene? And how would I know that one was different from the other?
it Good question.
Well,
um
Yeah, I'm still a little partial to the view Sharon is bringing up that scenes are different and that they are always there. And another thing I like to say is there's something that we operate within rather than something we operate on, right? So in the case of going to the bathroom, like I operate within scenes to go to navigate to the bathroom. And if instead the task is to, pick up the cup, I operate on that cup. Um, so yeah, I do think at some level there, maybe the cognitive map, like a set of cognitive map instructions to go from get to the bathroom or get to the cup could be described in a similar way. But eventually there's an operation that is either within or upon that is very different between those classes of stimuli.
maybe. Do I operate within or, well, I don't operate within an elephant, but I'm not sure I typically operate upon an elephant either. Though if we get biblical, you could have the Jonah and the whale. Is the whale a scene or an object?
a scene, well, both actually.
This sounds like a splitter lumper kind of argument to which the answer is undoubtedly both, but
Well, a car is also in that category of a
oh
whale, to
yeah,
be
that's
honest.
maybe better than whales.
Yeah.
All categories are more, two distributions with some overlapping, areas. But I think the vast majority of objects are things that we operate upon rather than within. And it's rare that one is within an elephant.
Yeah.
Though an elephant perhaps can become a scene in some cases.
And it's a really tough experiment to do inside a scanner
It
because it's
is.
really
Yeah.
tough to get that elephant into the bore of the scanner.
Hmm. And to get the NIH to fund it.
Um, Caroline, I want to thank you so much for joining us today. It was really a real pleasure and really interesting talk, wonderful science. Thanks so much for joining us. And
Thank
I also want to say that next week we have Mi Young Kwon, if I'm saying this correctly. She's going to be presenting her studies. And yeah, I hope you all have a lovely continuation of this week and not too cold. And thank you so much again
thank
for
you.
this
Thank you
wonderful
all
work.
for the great questions. And yeah, would love to talk more.
Yes. Ciao.